started, and people can join in. So I was going to go over some of the experimental design considerations. So you've gone through the physics behind the DNP, you've gone through the hardware that's required for the DNP process, and the chemical probe design, and the pulse sequence design for acquisition. And so now is like how do we put together all these different parts in order to make it work for our specific biological question that we are trying to address. So I'm going to cover a few different aspects of it. The T1 applications of what are the different things out there, but not too much into the details of it. And I'm going to concentrate mostly on the metabolic applications, the different aspects of it that we need to take into consideration before you actually use it for your experiment. And then how do we go about testing it, some of the options out there, and then the choice of probe. And so I'm going to start off with T1. The T1 is the be-all and end-all of a given probe, right? So you need to have a long T1 in order for you to get substantial information as that probe is being metabolized in vivo. So at 3 T1s, there's only 5% of your signal, total signal remaining, right? So the rule of thumb that we usually use is you get up to 5 T1s, you get enough signal. And T1 is field dependent. For example, here the Pyruvic T1 at 3 T is about 67 seconds, 11.7 T is 48. So you can see for this C1 of Pyruvic, the carboxylic group, the T1 decreases with increasing field, which is not really a problem for us in clinical applications because they're all in the lower regime, right? But for other probes like DHA, preclinical evaluation of DHA whose T1 at 11.7 Tesla at 22 seconds makes it impossible to use our microimaging. So I don't think anybody has successfully done a DHA shot on a mouse on the 14T. You just don't see any metabolism. And also, we are only talking about the T1 in the magnet. But what about the T1 of the probe as it's coming out of the polarizer and you're walking it along to your magnet, right? And in the preclinical lab, that's something we've been dealing with a lot in the recent past, right? So that brings into focus the path or the behavior distribution going from the polarizer to your magnet. And that is because the relaxivity is vastly different depending upon your field. So here is an example. I've tried to put references under each of the figure. Let me know if it's not obvious. It'll be up on our HNTRC website. So here you can see the relaxivity. This is of acetate. So this triangular one is acetate without any radical. And you can see this starts from about 20 millitesla to about 10 Tesla. So you can see that it's pretty low, it changes with the field. Whereas with the presence of a tempo radical, you can see a drastic difference. So as you're going from the polarizer, you're going into zero field. If there's actually a reversal of the field, you walking your sample through a zero field, you will quench your polarization completely. So it's very important to know the path through which you're walking with the sample. And one way to overcome this, we've been using in our lab the magnetic carrier that was built by Hong. I don't have the reference in here, but let me know if you need the reference for that. And so once you add a good sense of the way the lab is set up and the path you need to take for all the samples, you can think about the different applications of hyperpolarized MR. Is the most esoteric one, I would say, is the receptor ligand binding. The example shown here is vials containing different concentrations of cyclodextrin. So the numbers in here are the concentrations of cyclodextrin. And what was added is hyperpolarized benzoic acid. So you can see with increasing concentration, you have reduced intensity. So this is a negative contrast. And so this is one of the reasons why it has not been very widely used for receptor ligand binding. It's because you get a null signal when there is more receptors. Any ideas why? Why do we lose the signal when it's bound? It's the very basics of NMR, the correlation time. Again, back to the T1. The T1 is completely quenched because the ligand is bound really tightly to your receptor, thereby increasing your correlation time. Okay, so this is a very cool application, but it's not very well used because of this negative contrast region. It's very hard to implement it in vivo, and so that's why it can never match up to the PET application, which is way more powerful. And so the most robust application that's very powerful is to study metabolism. And also in the recent past, the studying perfusion and permeability to understand the various different hyperpolarized molecules, both endogenous and otherwise, sheds a lot of information on the intravascular, extravascular spaces much more easily. And it's easier to model and visualize in a sub-second type time scale, and also in terms of the use of urea, which is has a much better patient profile compared to contrast agents using gadolinium. So I'm going to focus mostly on the metabolic applications today. And so this is a very nice review by Miriam Chaumet, and this is a flow chart of how do you go about selecting a probe. What are the stepwise characteristics of how you make it into a successful hyperpolarized probe for your application. So the very first characteristic you want to think of is very rapid enzymatic conversion. It should be quick transport across the membrane. That would be the first step for the cell to take it up. And the example shown here is the classic example of why glutamine, even though in cancer biology, is a very, very significant substrate, as significant as glucose is for the cell to grow in very aggressive tumors, is not been a successful probe. Is that because its uptake is very slow. So the black bar graphs are that of glutamate signal, and the white is glutamine. So as you can see in the live cell, meaning the cells are burst and there is no more membrane for it to cross, you get almost a 1:1 ratio of your conversion of glutamine to glutamate via GLS, glutamine synthase. And then as when you have an intact cell with the increase of various substrates or additives, the glutamate production is reduced. And so this goes to show in an intact cell, the transport for glutamine is the limiting factor. This could be an artifact of this specific cell line under study, but in general, the ASCT2, which is the transporter for glutamine uptake, needs to be really, really high for this probe to be good and it works well. And so the third aspect that we just saw, like the benzoic acid example, is preservation of the polarization during the biochemical reaction. So here's an example again of salicylic acid and ascorbic acid. In just that in solution, you see really sharp peaks. But once you add serum albumin, which is a really large molecule protein that non-specifically binds to a bunch of small molecules in the blood, and this is the most abundant protein in the blood, and it can bind up to 50 different molecules, and it can take as many as like 20 different molecules at a given time. You can see that the signal is again squashed. And so you can see, again, the same concept of the tight binding, increasing of the correlation time causes the T1 of these molecules to become very large and the line crossing. And you see the reduction in the signal. And another unique aspect that we've seen is the metabolic food cycles. So the open squares are signals of hyperpolarized lactate in the liver, the vasculature is the square, and then in the kidney. With the addition of just 80 millimolar lactate, no C13 label, along with your hyperpolarized pyruvate, bumps up the signal in every single probe. So this has been one of the basic underlying themes, at least for pyruvate and lactate, is that the lactate signals you're seeing is not necessarily the net production of lactate, right? It is mainly an exchange. Can anyone tell me what exactly is the exchange here in this scenario? Is it the C13 molecule, actual nuclei that's being exchanged? What do I mean when I say it's exchanged? It took me a long time to understand that because, you know, C13 molecules, unlike hydrogen, does not move from molecule to molecule because that's the basis of a robust hydrocarbon or the basis of life, right? Because carbon forms really strong bonds. That does not undergo like the hydrogen exchanges from molecule to molecule. So what do I mean by exchange here? Any ideas? Polarization doesn't really transfer from, so if you, that's a very good idea, like the magnetization transfer, right? But here is the actual back reaction. When I say with respect to no net production of lactate, right? So for every hyperpolarized pyruvate molecule that's being converted to lactate, a thermally polarized lactate is being converted to pyruvate. Right? Because the LDH enzyme, which is responsible for the pyruvate to lactate conversion, works both ways. It's not a unidirectional enzyme, right? Most enzymes are not. So that is what I mean by exchange, is because the exchange between the enzyme reaction pool, between the thermal and the hyperpolarized. And so this is, again, inherently, that means that the product that your hyperpolarized probe is going to be converted into, that product concentration pool should be higher for you to be able to see a good signal upon metabolic conversion of your probe. So the classic example of why hyperpolarized pyruvate is such a big success is it meets all these criteria. Long T1s, very quick transport across the membrane, very rapid enzymatic conversion. It's not a tight binding. It's a very loosely bound hydrogen bonding between the substrate and the enzyme, and it preserves the polarization because of these. And this has been applied across the board for every application that you can think of, like oncology, cardiovascular diseases, diabetes, inflammation, you name it, and it's been applied. And so once you have the different aspects of the probe all worked out, now you want to test it. So any ideas? Where would you start? Would you just directly inject it in the animal? How would you go about testing it in a biological setting? Where would you start? Yeah. That's where I would start. So the very simplest way is, if it's an enzymatic probe, you would, if the enzyme is not too expensive and it's isolated, you could do a simple calibration study of the enzyme in various concentrations, like we saw for the cyclodextrin example. Or we then move to tube studies, is you lyse the cells, right? But then remember the glutamine example we just discussed. If you lyse the cells, you remove one big hurdle that might actually come back to bite you when you go to the in vivo system. It's the transport across the membrane. You lose that. But this is, again, a very, very quick test to see whether or not your probe works. It could be a quick test to check if the enzyme concentration in your particular cell line or pathology of interest is large enough. But there are actually, in my mind, the cell lysis experiment is way more difficult than an intact cell experiment. Any ideas why that might be? Think about the basic biochemistry. What do you need for an enzyme? The different cofactors, right? The minute you lyse your cell, the protease is going to chew up all of your cofactors, the ATP and everything. And then you need to think about adding those back in before you actually do your cell lysate or add the protease inhibitors to that system, which might end up non-specifically binding to your probe and cause further complications. And so the easiest way would be to do cells in suspension or pellets. But if you want to do very robust repetitive experiments where you want to quantify the rate of kinetics of these enzymes, I would move to my favorite tool, which is cells in a bioreactor. The reason I'm going to go into this because that's the bread and butter of what I do, and I think it's a very, very powerful tool to understand the biology of a lot of what we study. So this is just a very simple heads-up comparison between doing experiments in a suspension manner versus a bioreactor. First off, one thing that always wins over the bioreactor setup with a cell suspension is higher throughput. If you discount the time and effort needed to grow up the cells required for each experiment. So you need fresh sample for each hyperpolarized experiment that you want to do for each replicate, which means you need as many as a 30 million cell minimum for a 5 millimeter tube experiment, or like 100 million cells if you're using a 10 millimeter tube experiment. Whereas we have done experiments with as little as 5 million cells in the bioreactor. We can use the same set of cells over and over again for up to a week. We can treat it and study it over a week. And so it's much, much easier to reuse, and we can very finely control the conditions. So that is the other big issue is when you're trying to study some new system or a new probe, you want to be able to see that your homogeneity is maintained as you drop the cells in the NMR machine so that you can identify or resolve the peak. And so mixing of the sample and the hyperpolarized media is an issue. So as you're mixing it and you drop it over a period of 5 minutes or how much ever your acquisition time is, cells are settling. And so that causes a varying homogeneity in the field. Whereas in the bioreactor, we have been able to achieve such high homogeneity. We've been able to resolve intracellular versus the extracellular signals, meaning we can study in real time the hydrate that's being transported into the cell, converted to lactate, and we can see the process of lactate equals. So those are some of the really nice features using bioreactor. But the setup and the bioreactor schema that each lab uses is different, and that part of it is not trivial. You need one entire day to do one bioreactor experiment for the setup. And so this again leads back to the intra-experimental variability, because the cells maybe something weird happens with your polarizer, and so the cells you trypsinized is sitting on the bench for 20 minutes, and then it's sitting on ice, so you don't want the cells to be metabolizing and getting hypoxic while you're setting something else up. And then you go from, let's say, 4C to directly to 35C, the temperature differences. So those are minute details, but those could bring in a huge variability over each replicate of your experiment, whereas those are not really an issue in the bioreactor. So that's just something to be considered when you want to do a little bit more delving into how your hyperpolarized particular probe behaves in a given system. And so this is just a little bit more details on what exactly a bioreactor is, in case you haven't seen it. It's just a microengineered perfusion system. Such a system has been in use for ages, even with NMR. So most of the bioreactors are used in pharmaceutical or cell culture type applications. But this has been used extensively for over 40 years to study perfused organs such as the heart and the liver in the NMR field. And so what we have in our lab that we use a lot is miniaturized version with 5 millimeters, where we can have really high homogeneity, and we can do serial dynamic multinuclear measurements. So I can check on the viability of the cells and then do a sequencing experiment, go back to do phosphorus, and look at the bioenergetics of the cell. So here is the 5 millimeter NMR2, and these red lines denote the RF coil, and this is a baffle that prevents the cells from flowing away. And so you need some way of keeping the cells in the RF sensitive region of the bioreactor. So this example shown here is cells grown on polystyrene beads. Basically plastic beads, which are like any size from 10 to 300 microns in diameter. The other setup that is routinely used in our lab is an electrostatic procedure of encapsulation of the cells into microbeads. And so we can vary the diameter of the beads by changing the voltage between there. So this syringe contains the cells mixed with alginate, which is chelated by calcium in this lab here. And so we can keep these beads around again for a week. We've done drug testing on these same beads before and after. And it provides for a very robust platform. Here is an example of using this to encapsulate just the LDH enzyme. And what this inset shows is we polarized pyruvate and urea injected into these alginate microspheres containing only the LDH enzyme, and you can see the lactate production. And what we show here is over time, when these beads are encapsulated with different concentrations of the LDH, you see increasing concentrations of the lactate being produced. And simultaneously, this is what we add to it, because NADH is a cofactor that's required for the conversion of pyruvate to lactate. We provide the NADH, and we see it being converted to NAD using what during that experiment. And to me, this is the most powerful application of the bioreactor, is to study living tissues. We've done this using both prostate and renal tissues derived from patients during surgical resections. We take a small 8 millimeter core, slice it using this special meat slicer into 300 micron thick slice thickness, and then we can put anywhere from 4 to 6 slices in the 5 millimeter bioreactor. And here's an example when we injected C1 and C2 labeled Pyruvic into the bioreactor, and you can see almost simultaneous, instantaneous production of lactate as well as glutamate. So you remember how this particular process, going from pyruvate to glutamate, is a 5-step enzymatic process. So the polarization is preserved at every single step. And you can also see the signals for the bicarbonate and a very, very small signal for carbon dioxide. So depending upon your choice of the probe, you can see multiple pathways. So just glycolysis or through the ECA. And you can see very robust signals. And this is from renal cell carcinoma patients. And so we've looked at all the ex vivo applications of hyperpolarized MR, but what are some of the considerations that you really need to think about when you're doing in vivo studies? So one big thing is, because this is a step just before clinical translation, this is a step at which you would test the toxicity of the probe, the different components that you're using to make your probe. Another big thing is the pH of the injection. This needs to be very close to 7. I think for the human situation, we allow anywhere from 6 to 6.5 to 8, which is a really large margin. In terms of the animal studies, once we get the animal on the table, we need to keep the catheter patent during the time that things are being polarized. During a failed shot and you're waiting again for the polarization, you need to keep the line patent. So you're constantly injecting loading the animal with heparinized saline in order to prevent blood clots and messing up your equation. So you cannot, these mice are anywhere from 25 to 45 grams, and their blood volume is usually, rule of thumb is again 6% of the body weight. So at any given time, you do not load more than 20% of their total blood volume. So during an experiment, you might be injecting as much as like 3 mils, which is like 50% of the blood volume, because you have to keep the line even. So we usually inject 50 microliters or 50 microliters for every 10 minutes, and the mouse is in there for 3 hours, and then you load 350 microliters for each of your hyperpolarized injections. And so all of that adds up, and so that is something, and we've lost animals because of that. So that's something you want to really consider and keep things as much as possible ready to go once the animal is in the scan. So another interesting work is the concept of, especially in the brain, is the depth of anesthesia. So just like your concerns about the anesthetic agents when you're doing fMRI type studies, if you're doing brain studies, you want to consider the varying levels of the lactate to pyruvate ratio in the different regions. So I think in the brain, they found a significant difference between the lactate to pyruvate ratio in deeply anesthetized versus slightly anesthetized, whereas there wasn't much of an effect in the lactate to pyruvate ratio, let's say in the kidney or in the liver. Another work that was published was the effect of fasting. And this comes about just before the clinical translation. And what's being routinely done for, let's say, a PET study is the patients come in fasted. So this group, Brindle's group, did a study between test recess measurements in fed versus fasted animals. And they found a much tighter correlation between the two repetitive measurements in the fasted state. But this might be a problem, depending upon what you're trying to study. So in this specific example, they were just looking at the lactate to pyruvate ratio. So this was like a prequel to their first clinical trial, where they were evaluating whether or not they wanted the patients to come in fasted. And I think that is the protocol that they are using there, the patients come in fasted. I'm not sure. So this might backfire on you. Any ideas why? I'll show you in the example next. It depends upon the very specific question, again, you're trying to answer. So another very specific requirements for doing hyperpolarized MR is you can pretty much do any kind of MRI experiment on any type of, let's say, I'm talking very specifically about a tumor application, a cancer biology application here, is that a solid tumor, a cystic tumor. But a cystic tumor for a hyperpolarized experiment is just not going to work, because you don't have enough number of cells and enough number of vessels to perfuse those cells in a cystic tumor. So not all tumors or cell lines are amenable to be imaged. So what most labs do is they have one tumor model that works for them, and that's what they go with. And we were trying to do something in the renal cancer space, and we went through 12 different cell lines before we found really consistently solid tumor growing cell lines. And so the basic requirements for this would be a well-perfused tumor and the volume of the tumor. So this kind of dictates where you want to implant the tumor. A lot of the work, again, going back to the renal cancer example, is we were doing an orthotopic model. And so we would implant it in the kidney, as opposed to a genetically modified model that we use, again, a lot in our lab, is the TRANS model for prostate cancer, where the tumor itself can be anywhere from 2 to 4 cc, whereas in the kidney, you're restricted to no more than 0.8 cc before the animal gets really morbid. So then that would then also influence your pulse sequence of choice. So I'm not talking about that aspect at all, because that is something what Jeremy already covered. So you know the pros and cons and the strengths and weaknesses of the different pulse sequences, and you can just adapt that by varying the imaging parameters for your specific application. And then another big reason for failure or the loss of an animal is the characterization. Without this, you can't do a hyperpolarized experiment with an animal, right? And so you have to be a good expert, first of all, with characterization. Fortunately, we have an expert here. And also, as the animals get big in terms of cancer biology applications, the tails become really, really tough. And so that, again, complicates things. Then your vascular function itself decreases, generally as part of cachexia or morbidity associated with the tumor. So those are things that that will dictate the time at which you can get your experiment, or the number of replicas or the mice you have to account for losses. And so the next thing to consider is, once you've nailed down the in vitro or the in vivo or the ex vivo system, is the probe of choice, right? So the most important, for example, for pyruvate, which I think is a special case, is your choice of your label. It can be in the C1 position, it can be in the C2 position, it can be C1 and C2, but not in C3. Any ideas why? Remember, Dave covered it in the lecture. Again, going back to T1. The C3 is a CH3, a methyl group, and so the T1 is going to be really, really short. And so that's the reason why we don't do anything on this C. And so, why does it matter? Can you choose C2 pyruvate for your experiments as well as the C1? So for this, you really need to know the biology you're trying to answer, right? So the very first step of pyruvate to lactate would work for C1 or C2, but your T1 between the C1 and C2 is going to be drastically different. That's why I put the T1s here. I don't have a specific value for the C2. I don't think I have it here, but there is a publication from last year where Celine Hadline has published for the C2, and I think it's on the order of 20 seconds for the C2 last year. And so that then diminishes your acquisition window to observe that signal. So if lactate is all you want, you would want to put C1 pyruvate to maximize your T1 time range over which you can acquire your data. But if you want to simultaneously measure not just the glycolytic stage, but also the further downstream metabolism of TCA cycle, then you would want to use either C2, C1 copal, or a C1, C2 dual labeled pyruvate molecule. Okay, so another choice that you make is dependent upon your organ, right? So if it's pyruvate carboxylate flux that you're interested in, which is heavily prevalent in the liver, then you can get away with C1 pyruvate, which provides you with higher T1s, and so you will better equipped to measure the other further downstream metabolites as opposed to the C2. And the pathology that you want to detect is what I was saying about glycolysis versus the PDH flows. And this is the example I wanted to give you. So if you look here, this is from McNair's paper. The top row is this is I think, a liver perfusion experiment where you can see in the fasted animal, this dotted line here represents that of the bicarbonate signal. So you can see the drastic difference between the fed and the fasted. You have much higher bicarbonate signal in the fed animals versus the fasted. So if you were to, what we talked about previously, is if you were to fast your animals, you will not observe any bicarbonate signal in your experiment. So you need to be very careful about the multiple different parameters and what you want to measure. or misinterpret your data. If you don't see bicarbonate, then you're missing a key component of your PDH enzyme, because a lot of work has been done to show that the bicarbonate signal here directly correlates to your PDH activity. And so there's been a couple of papers by Matt Merritt who've shown that, especially using this refuse reverse system. And so another powerful application is doing multiple substrate polarization, so multifold. The few things you want to keep in mind when you want to do a couple of different substrates to polarize at the same time to give you different information is spectral overlap. Make sure the two are well resolved, and so are its products from that. If they overlap, then it means the purpose. And this also has some implications in terms of your acquisition parameters, how you're limited. If the two substrates are more than 100 or about 100 ppm apart, given the carbon spectral width that we have, then you're going to have a lot of overlap signals that's coming in the window, and so you should be able to resolve that. But you can get concurrent readout of, for example, the most common copol that we use is pyruvate and urea, where you can get information from metabolism as well as perfusion. And this is just a simple example that we did in a one-off experiment to show you can measure metabolism and redox at the same time. So this probe, dehydroascorbic acid, is a probe that gives you a readout of the glutathione and thereby the redox capacity of your cell. The DHA is converted to vitamin C and pyruvate is converted to lactate. This was done in a PDX model of renal cancer. And so here are some applications, very specific applications of C1 pyruvate, just to show you how powerful it is. Even though it's just a simple glycolytic readout, a single step, you can get the lactate that's converted from pyruvate. The signal gives you a very good readout of the tumor grade. We've shown that in both renal cancer as well as prostate cancer. You can get use this as a biomarker of treatment response, and this has been shown by numerous publications from brindle groups, from radiation, and as well as chemotherapy. And I think there are 5 different groups who've done work on radiation. And we showed that actually hyperpolarized metabolism, by just virtue of the multispectral readout, is more powerful than PET. So here we did a NAPT inhibitor of simple PCC cells. So what it basically does, it removes the cofactor. And that has been one of the therapies that's been tried, although unsuccessfully, for cancer treatment. And what we showed is that in the PET setting, the decrease in the lactate signal and the FDG signal correlated very well. But we also had an additional information from the hyperpolarized data is the much more increased production of the alanine signal with a concurrent decrease in the lactate, showing alternative pathways or escape mechanisms of the cancer cells that we could further attack in combinatorial therapies and things like that. This has been applied to look at inflammation. I think there are like 5 very key publications in this field. Genetic markers of IDH mutation. The Renuka lab has done a lot of work on looking at very specific readouts of gene-based mutations for brain tumors. And the biggest application is the ultimate clinical translation and application in 4 different pathologies now, totaling over 18 trials across the world. And here are very key examples of successful probes that has been actually used in vivo setting. So we've gone through a bunch of information. So I wanted to end with a thought process question. This is an example of using a few different probes to look at pH. So there are multiple probes available. pH on a very key characteristic of cancer biology. most aggressive tumors end up having extremely acidic interstitial pH, causing therapies to fail. And it promotes metastases and more proliferation of the tumor. And so pH is a very important metric, but very elusive parameter to measure in vivo. And here, there are currently 4 different in vivo applications that have been shown to measure pH. One of them is hyperpolarized pyruvate by the UPenn group, Dr. Rizzi's group, where they have decarboxylated hyperpolarized pyruvate very rapidly using hydrogen peroxide to make hyperpolarized bicarbonate and inject that into the animal after recovering the hydrogen peroxide. You don't want to inject hydrogen peroxide into living things because that's going to gobble up your redox and ultimately kill the animal. And this is cesium bicarbonate polarization, direct polarization of bicarbonate. And this is done by Rob Flevel's group and Dave Parentian, where they hyperpolarize glycerol carbonate, which is again decarboxylated right after polarization, and then the hyperpolarized bicarbonate is injected. So all 3 of them deal with just hyperpolarized bicarbonate, but to achieve that using 3 different substrates. And the bicarbonate and carbon dioxide are in equilibrium, which reflects the pH for a given pKa. And I think the pKa in vivo is about 6. And so here, this process is an extremely fast process, aided by this enzyme called carbonic anhydrase. And incidentally, carbonic anhydrase is upregulated in a lot of tumors. And so this forms a very, very convenient and easy to read probe. And so you have 3 different options. And the 4th one is a more recent publication using zymonic acid. Here is a subcutaneous tumor, and this voxel here is, the spectrum is from this voxel here. And this zymonic acid, they deconvolved this one peak into 4 different peaks of varying pHs. And so the way this particular probe works is based it's a chemical shift reagent. This zymonic acid looks similar to the dehydroaspartic acid in structure, and so it has 2 protons that are has a pKa going from I think 4 to about 10, and so that is why you can you get 2 different peaks that can capture the range that's biologically relevant for an acidic or a basic pH. And so you can see here there are like this particular ZA1 zymonic acid peak 1 and 2 spans of about 2 ppm. And so based with this minimal information, which one would you choose? Or what are the few key things you would want to look at without really going into the details of anything? How would you go about if you want to measure pH using hyperpolarized in your specific tumor system, for example? What is the first few things you would want to look at? But that's a good point. So let's say toxicity. But then I'm showing you all examples from in vivo. So let's assume that the animal survived. This may be not relevant here, but toxicity is again a question that you want to think of for the clinical translation. Where you can't inject cesium, you can't inject hydrogen peroxide in people, and the filtering process of those specific elements may not be as trivial as we would normally otherwise do for a preclinical setting. What would be the second thing that you want to think of?
Would the concentration that you inject like affect the equilibrium at all, or would it just rapidly explode?
So you're first injecting into the blood. By the time it arrives at the tumor tissue, that's an excellent question. That is the very first question. anybody and everybody asks when people are coming up with these pH probes, because the buffering capacity of the blood is huge. So first it goes through the blood and it's buffered before it gets to your site of interest. And so, but that is a very good question considering the amount of hyperpolarized substrates that we normally ingest. You don't want to alter the equilibrium, the buffering capacity. You shouldn't meet the buffering capacity of your system then. So, but that's a good leading point to what would come next, right? So, associated with that is the concentration. Maybe I will send the questionnaire like I did last time, but I put this question and then one other question for you guys to answer. So I just open up the Slack. I just put it on the hyperpolarized MRI. So the first question just pertains to what we just discussed here.